Anatomy and physiology. We are here today talking about the juxtamedullary nephron. It is Tuesday, May 5th, and we're just going to jump right into it. So the juxtamedullary nephron is referring to the location where it's found. And if you remember from our previous notes that the majority, 80% of nephrons are located in the renal cortex of our kidney. So if you remember, the kidney has the outer and the inner portion of it. So this right here is called a cortical nephron, whereas most of the nephron is found above that little line where it separates the cortex from the medulla. What we're going to talk about today is the other 20%, the juxtamedullary nephron. And you can see that this long loop of Henle dips down into the renal medulla, and it makes for some unique absorptive properties of that system. So our notes today are going to all be on page 5 of your study guide packet. My picture, and if you printed a color version, it's already kind of color coded for you. But I would recommend in having 3 different colors on this page. It's red, blue, and green to represent the 3 different processes of reabsorption: the active, passive, and then the secretion, which is an active process of reabsorption into our tubules. And so you might take a minute to even kind of outline or highlight each of these little arrows in the proper colors. And then the text that flies in, I'm going to have it color coded as well. You don't necessarily have to write it. You could highlight it or just highlight the arrows. But it's good because the colors here indicate to you what processes here are active and what processes are passive. So this is our juxtamedullary nephron. You could even label that up here at the top. I don't have that labeled, but that'd be a good thing to put on this page. And let's label those structures we're already familiar with. So number 1 up here, this is the BC, the Bowman's capsule, which is holding in it that network of capillaries, the glomeralis. 2, because the arrow is going in, we can assume blood flow is flowing in through that vessel. So that's going to be my afferent arteriole. It's not labeled, but this guy leaving would be the efferent arteriole. This whole region, this section right here, is the proximal collecting tubule. And then we drop down and follow our loop of Henle. That's number 9, is referring to this whole thing. I want you to note, I'll bring this back up, but notice the diameter of the tubule changes right here. It gets really thin. And then about halfway up the other side, it thickens out again. That'll be significant later. And then we have this section, the DCT, our distal collecting tubule, because it's further from my Bowman's capsule on this end. And then the final piece of this nephron on this side is the collecting duct. Okay. Keep in mind, there's lots of blood vessels wrapped in and around all of these tubules. We labeled our blood flow previously. So all of these arrows, like where they're going and what are they coming out of, is blood vessels that are intertwined with this whole tubular structure here. All right, so let's talk about how we form urine and what is in it and what isn't in it, which is very important. One very common thing doctors do when they're diagnosing, let's say somebody is maybe diabetic or having some other underlying conditions, is they do a sample of the urine to see what your kidneys are filtering out, because the main job of all of this is to keep the good stuff in and pull the bad stuff out and put that in our urine so we excrete it. So our waste, toxins, things like that all get out, but we want to keep all the good stuff. Okay. And so that's what the focus of today is. Like, what's the good? What's the bad? What do we want to keep? What do we want to get rid of? So at the very beginning here, at the Bowman's capsule and the glomeralis, we produce filtrate. Okay. And filtrate is a passive process, meaning there's no ATP required. And the way that filtrate is filtered through the glomeralis is pressure. So I've mentioned this before, and I had you draw this when we sketched our little quick sketch of a nephron, is the afferent arteriole is a larger diameter than the efferent arteriole. So it's going from a large tube to a smaller tube. And because when you're forcing things from a large to a small, it increases pressure. And so where the pressure increases here is in the glomeralis, and the Bowman's capsule is there to capture all of that filtrate. So it's really teeny tiny here, but if you can write the word filtrate, this will pop up in a minute, but we're going to put that in this little tube right here. So what's in filtrate? Basically, it's your blood plasma. So if you can remember back to the blood unit, everything that we get out of our body via blood is, the blood plasma is going to be in this filtrate, except, and this is important, except for proteins. Proteins are too big. They can't get forced out. Red blood cells, white blood cells, too big. They don't get forced out. But everything else that's in our blood plasma, so water, salts, glucose, amino acids, I'm going to list everything here. All of that ends up in our filtrate. So water is one. Okay, we know plasma is mostly water. Bicarbonate, HCO3, that's the byproduct of our respiratory system. Okay, one of those things that's really important for the oxygen and carbon dioxide exchange. Okay, bicarbonate is in there. Sodium and chlorine, okay, or chloride, excuse me, sodium chloride. So Na plus and Cl minus. So these are ions, our salts, and these are all over the body, super important for sending nervous signals and all sorts of other stuff in our body. Then we've got hydrogen ions. This is basically what makes things acidic, okay, in your body. And we like hydrogen ions because it bonds easily with other things, and it's actually kind of important to keep in our bloodstream. Urea, creatine, and uric acid. These are the bad stuff. This is the stuff that's a waste product from our regular metabolism. We labeled on page 10, the very back side of your packet, what each of those are produced from, right? So urea and ammonia are from protein metabolism. Uric acid is nucleic acid metabolism. And then that creatine is produced through our muscle metabolism. And so these are waste products that we want to get rid of. But this stuff, I don't really want to get rid of. And I especially don't want to get rid of my glucose and amino acids. But all of these things listed right here are small enough to get forced out in this high pressure system. And that is what turns into, and I want you to write that down, filtrate. And that is what is in this tube here, is the filtrate. All of these guys. So I want to keep the good. I want to get rid of the bad. On this list, the bad here is E. Okay, and depending on how much sodium ions I have in my body and chloride ions, some of this is bad. Some of it gets reabsorbed, some of it doesn't. It just depends on your body's current needs. But most of it is salt. I've never tasted urine, but I hear that it is very salty because we do excrete out excess ions and salts, this sodium and chloride. All right, so we'll see how that all happens and plays out here. So each one of these little arrows is a different substance from the filtrate that's getting either actively or passively reabsorbed. So again, when I say reabsorption, so this first little arrow, 3, is representing the reabsorption of sodium and chloride. And that's an active process because they're ions that has to be using ATP and pumping them out of these the cells that are lining those tubules. All right, what else were you doing here? 4, bicarbonate, because it's also an ion, a charged ion, we need to pump it out actively using ATP to get it out. This is all happening in my proximal collecting tubule. Water is passively passed out. It goes from high to low concentrations. And so there is presumably a high amount of water in this filtrate because it's just been filtered out. And so water just is going to diffuse out through osmosis into our renal cortex. By the way, if you notice the little line here, so this is the renal cortex and the medulla is down here. All right, and then the other thing that we can actively pump out, and this is the best stuff that we really, really want to keep, is glucose and amino acids. All right, now this process of filtration doesn't get rid of everything bad. So in this case, we need to secrete from our bloodstream into the filtrate actively things like drugs, okay, things that are harmful, and some acid. Okay, so hydrogen ions that maybe don't get forced out because they're too tiny. All right, so these are going to be actively secreted into the filtrate in my proximal collecting tubule. So I have reabsorbed a lot of good stuff. I have made sure and take out any excess bad stuff, but I'm not done yet, okay? Your kidneys function, I think, in a day to filter, or at least passes through the kidney, is 180 liters of blood. That is a ton of blood. But what ends up in urine is only about 1.8 liters. So we reabsorb nearly everything that gets filtered out. And so basically, yeah, so there's a huge amount of reabsorption that has to happen. So now we're getting to our loop of Henle. And in plan book, I've linked an article from Khan Academy if you want to read in detail about this. I'm going to get into more detail on this later as need be. But this is kind of a pretty cool mechanism that your body uses and why there's a loop even there to begin with. So the loop of Henle is mostly meant there for reabsorbing water. If we were to actually urinate out all of the water that passes through our kidneys, you would be urinating all day long, right? Our bladders would have to be huge. We'd be storing tons and tons of water, and we would also have to be drinking tons and tons of water to keep up with how much gets filtered out. So it's really important to maintain your water balance. And so the loop of Henle is really good at this. So water gets removed from the tubules. And the reason is because what ends up happening here is a hypertonic environment occurs. So the area surrounding these cells becomes hypertonic. And when something's hypertonic, there's less water. And so water wants to flow to the area where there's less water concentration. The reason there's less water is actually because the sodium and chloride, these salts, are being pushed out of the loop of Henle as well. So basically the water is following the salt out of the loop of Henle. And so they call this whole process a countercurrent exchange. I think the article refers to it as like a countercurrent mechanism. But basically this water and this sodium and chloride ions, they work together in this narrow section of the tubule to make a hypertonic environment. And this actively forces out a whole bunch of the water. And the reason why this loop is long and drawn out and not so convoluted is to extend the time in which the filtrate is in this tube so that you can get out as much sodium and as much water as possible. So these two are tied together. And again, this is a passive process. Our hypertonic osmosis is non-energy requiring. It's just moving things from high to low concentration. So this doesn't require energy, and we absorb a lot of water here. In fact, probably the most water gets reabsorbed in the loop of Henle. Now, at the ends, notice that the tubule gets a little bit thicker in diameter here. This is an active process where any excess salt and chloride ions are going to get actively pumped out as well, making this even more hypertonic. And so more water will be leaving in this section, which is the medulla. So our medulla of the kidney is where we have this hypertonic environment. All right, so as the filtrate moves up and around, we get to a distal collecting tubule. There's way less going on here. One thing that the arrow is lacking, you can add a passive blue arrow here to show water gets reabsorbed even more in the distal collecting tubule. Water gets reabsorbed everywhere. Right? And then we have more sodium and chloride getting reabsorbed, and then any other excess drugs and toxins that didn't make it out over here get actively pushed out here. So we're basically double checking, making sure there's nothing harmful in our bloodstream. We want to use our kidneys to take care of all that and get it out of our bloodstream. All right, so now we're getting close to having what is urine left in this tubule. Notice that urea, creatine, and uric acid was never touched in this entire process. That bad stuff stayed in the tubule the whole time. I got my good stuff back, okay? Any other bad stuff, the drugs, okay, and the hydrogen ions, okay, they all got absorbed into the tubules as well. So now I get to my collecting duct, okay? And, before I get there, I'm sorry, there's some hormones that act on this. So aldosterone, which we've mentioned before in our endocrine system, it acts on the kidney tubules to increase our absorption of sodium and chloride here. And so the aldosterone specifically is acting upon this section of the nephron. And so that's why I want you guys to put that box right there specifically. It acts on. And then there's another hormone that acts on the collecting duct. That's ADH, antidiuretic hormone. So if you remember, ADH is what conserves your water. And this is what alcohol inhibits. And so somebody who has been drinking heavily is going to urinate more. We talked about breaking the seal before. And that's because this doesn't exist. And guess what we're reabsorbing in here is water. So we've got water, some more salt, potassium. We haven't mentioned this guy yet, but it gets actively transported into the tubule. Any excess potassium that we don't need, if you have too much in your body, goes into the collecting duct. And then at the very end, even more water gets absorbed. Now, you might be wondering, why is urea is a bad thing? Why are we putting that back into our blood? Only a little, but some of that urea goes back in because it helps the water at the very end to get reabsorbed even more. The whole, notice water is reabsorbed here, water is reabsorbed here, water is reabsorbed here, water is reabsorbed here, and then again at the end, we want to conserve as much water as possible and pull it out of our kidneys. Now, obviously, we urinate out water, but we absorb as much as possible in this entire process because we like water in our body. We want to keep it in there. So the urea basically just helps that water to get reabsorbed. So then lastly, step number 21, what is produced is urine. And the pH of urine is very similar to the pH of blood. It should be in the range of 7.35 to 7.45. There's a urine lab that we don't get to do, but in that you are going to be testing urine and figuring out, maybe diagnosing some issues depending on what was found in the urine versus, what's the pH and things like that. But the normal pH of urine is somewhere in the range of 7.35, 7.45, almost neutral, but slightly basic. Just a slightly basic urine content, pretty close to neutral though. And the reason why urine is yellow is because one of the things that gets filtered out amongst all these things is a pigment called Eurochrome. And just like melanin is a pigment that we produce in our skin and our hair, Eurochrome is another pigment that's in our body, and it's just a yellow pigment. It's also the pigment that's associated with like bilirubin. It's something produced by it's like a form of that. It's not this exact same thing, but it's similar. All right. Last little bit I want to mention, so this is really zooming in. If we were to zoom in on what's happening between this tubule and the surrounding cells and those capillaries. So here's that peritubular capillary. These are the little twisty guys that are all intertwined with the collecting ducts. This is the, lumen, the space, the whole of the tubules. It's yellow, representing this is where the urine is in our body. All of this stuff, when I talk about reabsorption, all of that stuff is trying to get back in the bloodstream, right? Our red blood cells are here, right? We've got some intercellular space. And the lumen is lined with these simple columnar epithelial tissue. Just like in our large intestine, look at what they have on them, microvilli. And when we see microvilli, we know it increases surface area. When we have an increase in surface area, it allows for more absorption. So just like in our small intestine, we have large microvilli. We have microvilli in the tubules of our nephrons to increase absorption of all that good stuff. It's not important you know how each of these get absorbed, but some of them can pass in between the cells through those tight junctions. Some of them just go straight through the cells as well. But basically all of that stuff is getting forced into our bloodstream, our capillaries here, trying to reabsorb all the good stuff that was in the plasma. All right, that's it for today. And your homework assignment should be pretty simple using those notes. Let's see if there was anything else. I don't think so. That I wanted to go over with you guys today. Message me if you have any questions or concerns. Happy Cinco de Mayo. See you.